Hello everyone, welcome back. Today I'm here with the second part of this questionnaire, and in this part I will discuss some questions from plant anatomy. Let's get started. So the first question is, nitromonas converts. Our options are ammonia into nitrite, nitrite into ammonia, nitrate into free nitrogen, or nitrite into free nitrogen. So its right answer is ammonia into nitrite. जो नाइट्रोमोनास होता है वो कन्वर्ट करता है अमोनिया को into the nitrite. So ये जो नाइट्रोसोमोनास है, this is the bacteria which converts ammonia into nitrite again. Then further oxidation of this nitrite gives rise to nitrate, and this is the form which is absorbed by the plants. ये जो form है, this is rarely available for the plants और plant इसको absorb करते हैं. That is nitrate. इसी फॉर्म को प्लांट्स एब्जॉर्ब कर पाते हैं, एंड दिस कन्वर्जन ऑफ नाइट्राइट इनटू नाइट्रेट इज carried out by this bacteria that is called nitrobacter, and here oxidation process occurs. So this is how these microbes helps in nitrogen fixation. If we talk about nitrogen fixation, so there are different ways of nitrogen fixation. Firstly, we have biological nitrogen fixation, then industrial electrical nitrogen fixation. So mostly we discuss about this biological nitrogen fixation. It is done with the help of some living organisms, especially some microbes and e.g. that contain one enzyme that is called NR, that is nitrogenase reductase. In case of industrial nitrogen fixation, it is done with the help of process that is known as Haber-Bosch process. So this is the process which is involved in atmospheric nitrogen fixation. By industrial means, then electrical fixation. It involves thundering and lightning, so that high energy breaks down nitrogen molecule into nitrogen atoms. They combine with the oxygen to form this nitrate. So firstly, in case of biological nitrogen fixation, this ammonia is converted into nitrite with the help of nitrosomonas. Then again nitrobacter converts this nitrite into nitrate, and this is the form which is absorbed by the plants. So plants uptake and becomes a part of its biomass. Then this is consumed by animals and it becomes part of animal biomass. So death and decay of plants and animals leads to ammonification, means formation of ammonia. It is again carried out by some microbes. This process is also carried out by some microbes that helps in disintegration or decomposition of living bodies after their death, and this process is called ammonification. Then finally this nitrate can be converted back into nitrogen, and this process is known as denitrification, and it is carried out by again some bacteria like we have Pseudomonas and Thiobacillus. So some examples of denitrifying bacteria are Pseudomonas, Thiobacillus denitrificans, then Serratia, then Achromobacter, and Micrococcus denitrificans. So these are some bacteria which are involved in denitrification, means conversion of this nitrate into nitrogen. So, and it is carried out with the help of water molecules. So this it results in one molecule of nitrogen and 6 molecules of water. This reaction involves formation of one nitrogen molecule and 6 molecules of water. So denitrifying bacteria are part of the nitrogen cycle, as they convert nitrates into the soil to free atmospheric nitrogen. This is called denitrification, and this reaction represents the half reaction of process of denitrification. The reaction can further be classified into 2 different halves, and each reaction requires a specific enzyme. And the transformation from nitrate to nitrite is performed by enzyme nitrate reductase. So the next question, immediately beneath the promeristem there is as. Our options are ring meristem, procambial strand, zone of determination, or ground tissue. I'm not sure about this question. I think its right answer is the ground tissue. If we talk about this promeristem, which is the embryonic meristem, it gives rise to primary meristem that helps in growth in length. So basically इसके नीचे present होता है ground meristem और ground tissue. So primary meristem, it is a type of meristematic tissue that is responsible for primary growth, and that primary growth is growth in length. जो length में growth होती है, that is called primary growth, and it originates from promeristem and produces cells that becomes permanent tissues. And so some examples of this primary or promeristem are ground and primary vascular tissues. An example of primary meristem is apical and ground meristems. So secondary meristem, it is the meristematic tissue that originates from permanent tissue, and this by the process of dedifferentiation, that is there are some permanent cells which regains power of cell division and they becomes meristematic. So that process is known as dedifferentiation, and after that they will divide again and gets differentiated. That is called redifferentiation. So secondary meristem is usually lateral meristem that helps in secondary growth, so involved in secondary growth and responsible for increase in thickness of the plant. So diameter को increase करता है, that is growth in girth. The next comes the difference between primary and secondary meristem. So this primary meristem originates from embryonic meristem, while secondary it originates from permanent tissues by a dedifferentiation process. The cell of primary meristem are generally rounded, oval, rectangular, while secondary meristem are generally elongated. This primary meristem have central vacuoles in their cell, while secondary meristem have this primary meristem have no central vacuole, while the secondary meristem have central vacuole. Primary meristem form primary permanent tissue, and they helps in growth in length, while the secondary meristem forms secondary supplementary tissue of the plant, and they helps in growth in girth, secondary growth. So the parts formed by the primary meristem are protoderm, procambium, and ground tissue, while this vascular cambium, this secondary meristem give rise to vascular cambium, cork cambium. They helps in lateral growth, that is growth in girth, also known as secondary growth. The next, this ring meristem. So this ring meristem is mainly associated with the formation of stamen clusters. जो एंड्रोशियम वाला पोर्शन है उसे बनाने में जो मेरिस्टम हेल्प करता है, that is called ring meristem. So this is mainly involved in the formation of the stamens in clusters, which are formed in clusters. The next, if I talk about the meristematic tissue and its classification, so there are different types of meristematic tissue on the basis of position, origin, function, and plane of division. So I will discuss in brief. Firstly, on the basis of position, meristem is apical meristem, intercalary meristem, and lateral meristem. Apical meristem that helps in growth in length of and is present in the apices of root and shoot, mainly involved in primary growth, while the intercalary meristem occurs between the mature tissue and responsible for internode elongation, while lateral meristem helps in secondary growth and that increases the girth. For example, we have vascular cambium and cork cambium. On the basis of origin, meristem is primary meristem and secondary meristem. Primary meristem is derived from embryonic stages and differentiated into primary permanent tissues. Secondary meristem, it is derived during later stages and helps in growth in girth, that is secondary growth. It produces cork cambium and interfascicular cambium, which are mainly meant for lateral growth. So lateral growth may involved with them, that is also called secondary growth, increases girth of the plant. Next, classification on the basis of function. So it is divided into protoderm that give rise to epidermis, stomata, and hairs, then procambium that give rise to primary vascular tissue, for example xylem and phloem, then ground meristem that give rise to all tissue except epidermis and vascular strand. Then finally, on the basis of plane of division, so meristematic tissue is of three types. One is mass meristem, it divides in all the planes. For example, we have endosperm, young embryo, sporangium. Then rib meristem or file meristem, it divides by anticlinal division in one plane, for example development of cortex and pith. Then plate meristem, it divides anticlinal into two planes, for example development of epidermis. So anticlinal division means the division which increases the number of. The next question is, tunica corpus organization is not found in. Our options are Saccharum officinarum, Zea mays, Ginkgo biloba, and Cycas revoluta. So this tunica corpus theory was given by Schmidt in 1924, and this theory is not applicable for this gymnosperm, that is Cycas revoluta, and all of and other options follow this theory, that is tunica corpus theory. So this theory was given by Schmidt in 1924 and is based on the plane of division. जो सेल का प्लेन ऑफ डिवीजन उसके ऊपर बेस्ड है. अकॉर्डिंग टू दिस थ्योरी, एपिकल मेरिस्टम ऑफ द शूट इज डिफरेंशिएटेड into two regions, there is tunica and second is corpus, and tunica is the outer portion of that apical meristem, and corpus is the inner portion of that apical meristem. So tunica, in this the cell divide only by anticlinal division. Anticlinal division means the division which increases the number of cells. So this division is known as anticlinal division, and it makes the epidermis, that is the outermost layer. Then next is corpus. So in this region the cell divides in all the planes, and it is responsible for thickness of the stem in young stages. So these are found in almost all the gymnosperms and angiosperms. Therefore, it is the most accepted theory. But Foster studied this theory in ginkgo biloba, so that is median hair tree, a living fossil, and showed that corpus is further differentiated into 5 regions, that is central zone, transverse zone, peripheral zone, pith, rib, promeristem zone. The next theory is histogen theory. This theory was given by Einstein in 1868. According to this theory, the meristem in the shoot is differentiated into 3 layers, which are called histogen layers, and the fate of these 3 layers is already determined. And these 3 layers are dermatogen, peribellum, pleurome. In this, the cell divides. In case of dermatogen, derma means skin, so it will form the epidermis, that is the outermost skin. Then peribellum, it will form the ground tissue. And here the division is periclinal, means that increases the number of layers. Then pleurome, it is the region. In this region, and the cell divided in all the planes, and it makes the stellar tissue, that is the vascular tissue. The next question is, the process of entrance of pollen tube through micropyle is known as. Our options are porogamy, charizogamy, mesogamy, or heterogamy. So micropyle के through जब pollen tube की entry होती है, that is called porogamy. These are the diagram which shows the entry of pollen tube through different structures. Like firstly, when the pollen tube enters through this opening, that is called micropyle, that is called porogamy. Then second is charizogamy, when the pollen tube enters inside this ovule through charizogamy, that is called charizogamy. But finally, we can observe from these diagrams that the entry of this pollen tube is through this micropylar end, because the egg apparatus lies near to this micropylar end. So that's why final entry is through this micropylar end. Then the third one is when the pollen tube enters through funiculus either or through integuments, then we use the term mesogamy. So porogamy, it is the most common type. और जो pollen tube है इसमें that enters the ovule from micropylar end. According to some worker, pollen tube is directed. Porogamy is directed by filiform apparatus. Filiform apparatus is a finger-like projection which is present in the synergids. Synergids के पास present होते हैं finger-like projections जो guide करते हैं pollen tube को. Next is charizogamy. Charizogamy is also known as basegamy. In this case, pollen tube enters the ovule from charizogamy end, for example, in case of Casuarina. Third is mesogamy. In this case, the pollen tube enters the ovule through funiculus or integuments. The next term is obturator. So this is again a very important structure, special structure, which basically facilitates the entry of pollen tube into the ovule. ये भी pollen tube की entry को facilitate करता है, help करता है to enter inside the ovule by forming a sort of bridge. And soon after fertilization, the obturator shrinks and disappears. तो fertilization के बाद ये disappear हो जाता है. The next term is heterogamy. In botany, heterogamous means when it carries at least 2 different types of flowers, that is male flower or female flower. That, for example, male and female flowers, or bisexual and female flowers. Stamens and carpels are. The next question, sapwood is otherwise called. Sapwood को और हम किस term से जानते हैं? Our options are duramen, alburnum, pith, or medullary. So its right answer is alburnum. So basically जो hardwood है that is present in the central region, जो कि dark color की appearance की होती है. It contains resins, tannins. Then the outer living portion, that is called sapwood, which is lighter in color and is less durable. This is called sapwood. So for sapwood we use the term alburnum, and for hardwood we use the term duramen. So there are certain differences between these two types of wood. First, sapwood is living part of the wood, while hardwood is dead part. And the sapwood is situated on the outer side of the wood and is less colored, that is lighter in color, while this hardwood is situated in the central part that we have seen in the previous image and is darker in color. This sapwood is soft in nature and tyloses are absent. Tyloses are basically the plugs which blocks the vessels or xylem vessels and tracheids. Then this hardwood is hard in nature and tyloses are present. This sapwood is not durable and is not resistant to the microbes, while this hardwood is durable and is resistant to the microorganisms. Then next type of wood is the autumn wood or spring wood. Basically जो autumn wood or spring wood had these both types of xylary elements are formed in 1 year. In spring, when there is plenty of food or water supply, then the xylem cells which are formed during this winter, they are large size and are lighter in color. While in autumn the conditions are not favorable, so the xylem cells formed are smaller in size and are darker in color. Thick word cells are formed. So both of these cells form a layer which is known as annual ring. So this is spring wood, this is autumn wood. So we can see here, so this dark color line represents the autumn wood and this lighter color line or ring represents the spring wood. And both of these wood types of wood form a single ring, annual ring. And by calculating these rings, number of these rings, we can calculate the age of plant. हम इससे प्लांट की एज का पता लगा सकते हैं, एंड द टर्म यूज फॉर दैट इज डेंड्रोक्रोनोलॉजी. Dendrochronology is a technique by which we can calculate the age of plant by calculating the number of these annual rings. So spring wood is formed during spring season and autumn wood is formed during winter season. And this spring wood forms the major part of annual ring because it is formed by large cells which are lighter in color, while it constitutes the narrow strip, this autumn wood. And spring wood is present in the beginning of the annual ring. It is presented at the end of this annual ring. Spring wood forms plenty of xylem vessels which are wider and larger in size, while this autumn wood xylem vessels are narrower. Xylem fibers are fewer in number in case of spring wood, while they are abundant in case of autumn wood. Wood is lighter in color as in case of spring wood and dark in color as in case of autumn wood. This spring wood is also known as early wood. The other term used for the spring wood is early wood and the autumn wood is known as late wood. So these are the terms which are used for these spring wood or autumn wood, and these are the differences. Next important term is dendrochronology. बेसिकली डेंड्रोक्रोनोलॉजी है, इससे हम कैलकुलेट करते हैं एज ऑफ द प्लांट, एज ऑफ अ ट्री, बेसिकली एज ऑफ ट्री एंड स्पेशली ट्रीज विच आर प्रेजेंट इन टेम्परेट रीजन वेयर इज कंप्लीट डिफरेंशिएशन इन द सीजन्स. वहीं पे हम इन रिंग्स को